So 2D doesn't work very well. The mice doesn't work as well. Same problem. I mean, we tried, okay, and it's not good. I mean, it's just, we are not mice. So there's clearly a need for something that's more relevant. You need to work with human cells, and you need them to be organized in something that look at least a little bit like human. Organoids, it's not a magical thing. You have to adjust it to a question. If you look at the progress which have been done in the last 20 years, I mean, everybody, for practical reasons, have been using basic 2Ds, doing automation, and it doesn't work. So you want me to say that they have to go to Cherry Biotech?
Can you introduce yourself and Gustave Roussy?
So welcome to Gustave Roussy. Gustave Roussy is that's now one of the biggest center it is the biggest center in France actually, and they claim to be in Europe too. So it's a cancer comprehensive center, which means we have it's a big medical facility, obviously, but it's also a big research center, and it's I think that's important because we have good contact with the clinicians, and we are really like an immersion, which is very important. I'm working in an Inserm unit. There are about 10 Inserm units in Gustave Roussy. And each unit is between 20, 30 people, 40 people, depends which one. And so I've been arriving in Gustave Roussy like 2019. Sorry. And so I started with this research unit actually. And my background is I'm a veterinarian, which is totally useless here, but which gave me a feeling of pathology, which was important, and which is one of the reasons why I came to Gustave Roussy, actually. I've been more like a basic researcher all my life as a scientist. And the idea was to get from the basic science to cancer. And there's a big gap between, actually. And Gustave Roussy was, for me, a nice place to be because of this contact with the clinicians, try to get really in contact with what's going on beyond, nice, fancy embryological models. So I've been working actually before coming here on the morphogenesis of the mammary gland in mouse mostly. So how you have tubers, which start growing during development and invading actually matrix to become an actual mammary gland. And naturally I came here to work on breast cancer because during breast cancer you have similar type of mechanisms. And I got mostly specialized in some type of a cell process which is involved during development and also during carcinoma progression. The cells during the progression of the carcinoma are going to change the phenotype, and you switch from a nicely epithelial organized cell to something which goes from, barely organized to totally individualized cells. It depends. You have all the situations going around. And that's, welcome to cancer. Everything goes basically. It's so be aware of, of the pre-built ideas. And so the idea was to see if you would have this type of process taking place, and if this could provide some kind of specific targeting. So when I arrived here, I started working. This unit actually specialized in immunotherapy, which was not really in the same line of specialization, but was also for me an opportunity, I would say, to discover something that was new. And that was actually very relevant to the type of work I was doing. Because when the cell changed the phenotype, it also changed, because it changed also, the antigen presentation. So it also changed the vulnerability to immune cells, basically. And the cancer cells typically become resistant to treatment, and they become resistant to immunotherapy because they change the panel of antigens, very simply. And this change of phenotype was really more like in my direct interest range.
When did you realize micro-physiological systems could be valuable for your research?
Well, to be honest, I mean, I'm working with 3D models since my thesis, which is not yesterday. And at the time, I mean, it's funny because, you see after when you have been in the business for a long time, hopefully you see a kind of a logic in what you have been doing. And I was working on the cell migration, and I was working on the morphogenesis of the thymus. Thymus also actually, input cells, which is funny. I'm working now people are working with lymphocytes. So anyway, so during development, there is a stage at which you have these hemopoietic cells, which are coming from bone marrow, wherever, it's not very clear. and they come through the blood, and they invade, which is something like which is an epithelial kind of a rudiment, which is going to become the thymus. And it's very weird because they arrive here and they get in, and they invade, really go through a basement membrane like a cancer cell. They go in, then they set in, and they become lymphocytes after a while, they differentiate, and a big story. So my point was really there was a time at which they go here and they invade, which was, kind of a reminiscent of a metastasis stuff. So at the time, of course, we did not have Cherry Biotech. But I was trying to design, some kind of 3D funny things with collagen and stuff like that. And very clearly, obviously, we had to get some in vitro models. And the first model I prepared, at the time, the prime was to get a basement membrane. This was before Matrigel, if you can imagine the time. And actually, what I used at the time was a human placenta. It's also another period, human placenta. So I was like a PhD student. I would go to the hospital and with a big plastic box and get my waiting outside from the delivery room, get the placenta and go with my bag, go back to the putting a plastic bag around so it doesn't show too much, and go to the lab basically and try to dissect it and get the membrane, and then put these hematopoietic cells on the top and see if they would be able to go through. So this was like that was a 3D model. That was but it was not very, very easy to do. It was very, we didn't keep working with it. When we published with it, that was it about. So that was the start. And the second thing is actually I was talking about Matrigel. I did my postdoc in the US at NIH, luckily before now. Nowadays that would be quite difficult. And I worked in a lab which was working on extracellular matrix. And I was absolutely not working with cells. I was working in molecular biology, cloning at the time at which we were cloning these genes.
Have you worked with animal models?
Not so much. I did work with animals actually, for the memory genesis. So I worked with these knockout mice that were not expressing the my gene of interest, which is involved in phenotype transitions, and which was a poor animal. A very poor survival. They had the face was kind of rough. Because it was involving the neural crest cells, which are responsible for most of the face. So these poor guys, they had the eyes were a little beyond, they had the short faces, and basically they didn't survive much. Anyway, so yeah, it was and otherwise here, because right now if you don't do organoids, you use mice basically. So what we have been using mice was for the pedics, for the patient-derived xenograft. So this I have derived some xenograft here, and that's useful as kind of an intermediary between the cell line and the biopsies from patients. And you need it. So lucky me, there is actually a platform in Gustave Roussy, so they have prepared them. So I just don't touch mice, very clean. This is kind of for now at least. We need a reference basically, and the cell line is. It's okay to work with, but it was not good enough for me.
What impact organoid solutions can have on cancer patients?
Well, that's very straightforward. If you look at the progress which have been done in the last 20 years, everybody for practical reasons have been using basic 2Ds, doing automation, and it doesn't work. That's sad. It doesn't work. It's, the ratio between the new molecules which have been found like this and the actual treatment is not good. Most of the improvements correspond to the fact that the clinicians are working hard, getting the right doses, the right combination, and the right, and in this direction, there have been certainly a lot of improvements, lucky us. But I would say that actually new molecules that have an impact that we can actually measure, it's very disappointing considering the investment in 2D. So 2D doesn't work very well. The mice doesn't work as well. Same problem. We tried, okay, and it's not good. It's just we are not mice in terms of cancer, and that's really. It's very useful for other things, other questions, but. And so there's clearly a need for something that's more relevant, and organoids can. It's interesting way. I think it has to do with the fact it's a little bit more complicated. And it's not as straightforward to do, large drug screening protocols. So I think that's why it has been so slow to come up. But I think right now it's really a requirement to see the field exploding everywhere international. I mean, there's clearly like now you have, I mean, you get every day, invitations to go to meetings, blah, 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 3D organoids. So of course, all organoids are not equal. And it's a new methodology, but I think in terms of, I don't see another way, basically. We don't have a choice. It's to know what a drug is actually doing at the level of the cell. As the 2D is not relevant, and the mice, you get limited, and you are not mice. So you need that. You need to work with human cells, and you need them to be organized in something that look at least a little bit like a tumor.
What are your ideal organoids and micro-physiological systems?
I think, organoids have to be, I mean, it's not a magical thing. You have to adjust it to a question, basically. So plenty of models are possible. And what is important is to be something that's relevant to the question. Are you going to focus on the microenvironment? Are you going to focus on the cell organizations? Um, Ideally, I mean, in tumor field, you want to do something that look like a small tumor, but even this is not simple because when you get biopsies, you get them from patients who are at a certain stage of cancer. You cannot ask them to come back 2 weeks later to get rid of the piece. It's bad enough as it is. And so you want to model something that's going to be at the start of the tumor progression, or you want to look at something that's more like a metastasis stage, and that's a different model. Every time it's a different matrix, different type of interactions, cell model. I've been working on breast cancer, and I'm kind of focusing on this stage where you have, in breast cancer, you have these tubers I was, trying to study earlier. So the tubers are still present, but the tumor cells are forming inside this tumor. So that's what they call the in situ part of the tumor. So as long as they stay inside, not too bad. I mean, you can, there is no risk of metastasis and stuff like that. And unfortunately, from time to time, they decide to get out. So they invade, they go through. And that's to me, I mean, for the pathologist first, it's really the moment at which you get a cancer, at which it, really gets dangerous for. And then they start going out. When they go outside, they reconstitute small aggregates that becomes bigger and bigger, and you have a tumor. That's really hard. So if you want to reconstitute this, you need to have tubers, you need to have, and it's complicated. If, on the other hand, you just want to see if a treatment is going to kill your tumor cells, take the tumor cells, put them in the collagen, see what happens. And I'm saying collagen, but if you put the collagen very stiff, the cells may use it to migrate. So what you will get is kind of embedding things, which is interesting in terms of morphogenesis, but which is not going to be the established tumor, which has like a gang of, not like a gang, like an overlaying, a big layer of collagen, which is going to be stiff, which is going to wrap around the tumor, and which is going to be like the reaction actually from the body against the tumor. Then you're going to have an invasive front where you have the adipocytes because it's breast cancer. You have a lot of fat cells all around, and you see the tumor cells invading. So that's another stage. So depends what you want to mimic in your model. So I think what is very important to me in this type of model is the fact that it's versatile. That you can do several things. You can change your environment. You say, okay, today I'm going to do it like this. Tomorrow I'm going to put some lymphocytes on the top, or not. Or so I want a system where that's you have different modules that you can adjust. Not like a large throughput model where you're going to get everything uniform in 2,000 wells. I mean, that's not the question I want to address.
How does Cherry Biotech meet some or all of these needs?
What I love in Cherry Biotech is, we have these 24 wells, so we have plenty of volumes. You can do whatever you want. You can put layers on collagen or not. You can put, So Cherry Biotech has been proposing and developing some models, which are nice, elaborate, and but what I like is, I can use a piece of it. I can do something else. I can put my own. When I mentioned I was using these micro supports, so I can put these micro supports in it or not. I mean, it's very versatile, and I follow. What is unique to me in Cherry Biotech is the ability to follow the fluids. So you can at every moment know what's going on by the metabolites that you're going to find, which are circulating through the chambers. So these are big hopes on the kind of thing that you can find there. The metabolites is one thing, so it gives you an idea. If you have lactates, okay, you know that the cells happy, glucose is going down, okay, so they're absorbing and so on. But you can also detect like little pieces of DNA. You can detect something that's very fashionable now, which is micro-vesicles that can be secreted by the cancer cells and be doing something else, somewhere else. You can look for circulating, piece of DNA, RNA, whatever, and cells. If you want to metastasize, you can catch them on the way. So I think that's a great thing. And I mean, yeah, in the system as it is now, it's easy to use. I mean, there is really, I mean, seen from this seat, it sounds like it's relatively straightforward. And okay, we're going to get to this, but the experiments we have been doing already show that it works. I mean, you need in vitro models to be able to see what's going on. So I think it's going to be more relevant with Cherry Biotech, because you'll be able to get a better microenvironment. So hopefully the results have more clinical, relevance. But yeah, you can use 2, 3, 4 drugs at a time.
Why is it important to control the microenvironment?
It's in terms of clinical relevance, it's really important because that's all these models have with 2D classic. You put a tumor cell line and then you kill them, but then we use the same drug. Well, you're going to kill the fibroblasts. Huh? I mean, yeah, it's just a matter of relevance. And but the observation, everybody is doing it. If you change the environment, you change the sensitivity to the tumors. Then it depends on the drugs, what it's targeted. So it's you need that to be specific to have an idea about what's going on. And so yeah, definitely microenvironment is a key, and microenvironment change between cancer types and between the cancer stage. You have this immune response, which makes that the tumor is not the same early on as it becomes with time. And the cells are going to become, you have this classic stages of response to the immune system. You're not going to get there. But what is important is it changes with time. So your microenvironment is going to change, and depends what you want to target. So whether this is the same. So yeah, it's crucial. I mean, in this very early preliminary experiments, it was like proof of concept experiments. Now we know. You put fibroblasts, well, different results. Why? I don't know. And that's why I'm here.
What can you do thanks to Cherry that you couldn't do before?
I can hope to see what the cells are going to secrete, to have an idea of what's going on. What I really like is the fact that we have real-time following of what's going on in the organoid, and that we can keep it for weeks. And usually when you do your little 3D collagen gel, it's okay for 1 week, and after 1 week things get bad. Have a harder time to. I could do my tubulogenesis thing for like 2 weeks maybe. And 2 weeks I had nice tubules growing. And after 2 weeks, actually, there is kind of a stasis. I mean, cells keep proliferating, but you can see that it's kind of more at the edge. And then the collagen, then you have the mechanical aspect. So here, because of the fact that it's renewed all the time, it's much more stable, and you can redo the experiments. You have a better way to. I mean, it's more reliable, basically. That's right. More reliable, long-term ability, and the ability to do something that looks like an actual treatment. When you do a treatment, you just don't put your drug in the cells and wait for 2 days to see what's happened. I mean, it goes for 1 week, and you stop, and the actual, and the protocol that's used for the patient is something that takes 1 week, 2 weeks, and is repeated. So this you can do it. I mean, which is to me is totally new. I mean, that's you can really get the organoids to get the same treatment as they would in vivo. And then you can get, because of the way the configuration, you have one well with a tumor, and the next well is going to be hepatocytes, is going to be something. So you can see the impact of the treatment on other cells as well. And then you can bring on the top some, of course, all type of lymphocytes, CAR T lymphocytes, all kind of engineered cells, or treatments, for the anti-PD-1 or whatever. So again, the versatility, the fact that you have like a Lego system, and you can, okay, today I'm going to put this and this, and make it for one question. In this case, it's going to be breast cancer, early stages, one specific subtype, and try to focus. Go from the situation where you have cell lines growing on a 2D to a situation when you have a luminal type B, early progression stage, model.
Do you have an anecdote of your collaboration with Cherry?
Beyond the crepe line? No, seriously, I would say I was touched when, the first time with this experiment with Cherry Biotech, and I had this PDX, these cells coming from a patient. Unfortunately, the story went bad for the patient. It was a very aggressive tumor. And to see these cells growing, in a macro environment for 1 or 2 weeks, that was kind of, you could feel that you were working on cancer, basically. It was a real contact with clinical problems. Don't know how to say it. But yeah, to me, it was very, very nice to see that we had the possibility to have an impact on the disease. And, it was just a matter of hard work. And
Okay.
so that was, I couldn't have done it, without a system like Cherry Biotech, definitely. And I would say what I find in Cherry Biotech, I'm talking with colleagues. I'm not talking with somebody who's trying to sell me a machine and trying to see what's going on and how we can get it better and then sort of something. And I say, oh yeah, but I read about this and this I love it because that's not something that I found with every company. Is to have some people who are at the same time, they have the background, the scientific background, and they're interested in science, in research.
Do you have suggestions to improve our Cubix machine?
Well, you can't have it all. What I enjoy a lot is the fact that it's versatile. I would love it even more if I could get still some more lines. Because right now, we have 24 wells and you have 4 lines. If we could have 6 or smaller. And I was actually contemplating, because everything is based on this 24 wells format, which is good because there is volume. Then if you had like a 48 format, would you get enough volumes? Because you would get definitely more lines. For me, in terms of tubing and stuff, that would be a real feast. But I think that would provide, because now we can have, like 4 duplicates is a bit short. So you can have, a second chamber. That's one way to solve it. So you get 8. I would say you need to have about 10 lines to be working. Not 200s, but 10. 10, so you can have a treatment, 2 or 3, 2 duplicates, 2 doses, have another one, a control, more or less around 10 samples, 10 wells, different 10 lines of wells would be good. But I'm not totally sure how to do it because on the other hand, the 24 wells is very easy to use. Everybody has it. It's, all the microscope platforms. So I don't know how. I suppose to have the 6 lines would be complicated for technical reasons because of still on the edge, I suppose. So maybe the way, what you guys have been doing, which was to use several wells, several plates, is a way to answer that. Yeah. So beyond that, using more biosensors.
Okay.
Yeah, because I say that's really going to, potentially, but we don't know. But being able to give the state of the organoid, saying if it's going good, bad, based on what's going out, it would be. That's a new thing. It's right now it's still, I think, like an exclusivity with Cherry Biotech, as far as I know. But I'm pretty sure a lot of people are going to do it around and being able to get to isolate, the same, vesicles, DNA, metabolites, and whatever.
Yeah.
And with all the omics going beyond, it's obviously something that's very, very rich in potential.
What would be the first sensors you would
Sensors?
that?
Well, still to be, but beyond doing, rather than doing this kind of toxicity assays where you use propylamine iodide or something like that, which are okay, but not very, very specific. You get to microscopy, to the problems of image analysis. If we could get something coming through, that would be so much, and if possible, specific for a cell type. And there is no reason we would not, if we get some kind of piece of DNA, if I could do like a fast sequencing of the little piece of DNA that goes through, and say, okay, I got a little bit of DNA, so that's some coding, like a gene that's specific for tumor cells. We know that the nuclei have been breaking up. I get some, of course, it's not easy to set, but that would be great. That would be really, so you could follow, then you don't need so many lines because you're going to do a small dose, see nothing happen, increase the dose the following week, like they do. So, back to you guys.
Okay.
How is the organoids and 3D cell culture field evolving?
Okay, you have to separate the research from the clinical use. I'm not so optimistic for the clinical use because of the pressure. I'm going to be of the market basically, because I can see how the forces which have been making big companies using 2D system for years and investing plenty of money and means to get this system. Even so, from the start, it was a big bet. And at the end, the bet is not so. The result is not so good in terms of financing and investing. Then you had like the PEDIX wave, and a lot of mice have been killed to try to get a good model, and it's not a good model as well. It does answer things. It's not it's very pertinent and good and relevant for some questions, but as a large screening tool, it's not good. On the other hand, in terms of for the research, the field is exploding, and there is no way to go back. All the research facilities are doing organoids. They don't have a choice. It's it's fashionable, but it's not only unfashionable. It's a new methodology that you have to include in your questions. The main problem with the systems now is the output. It's still very, very, it's really disappointing because most of the time, as what I've seen before, what you're going to see is the cells are alive or not. And that's not good enough to me. You want to know more what's going on. You want to see if they're suffering, if they become more sensitive to immune cells, if they're differentiating, if they start isolating, migrate more or less, what's going on in terms of the differentiation of the system. And that's definitely going to take some fancier model than the basic.
Which advice would you give to someone who is starting with organoids?
So you want me to say that they have to go to Cherry Biotech? No, I would say they will go by themselves anyway. But they have to try. Get your hand dirty. Try some matrices, and don't believe anything. Just try. Try for yourself. Try everything that you go around. Read a little bit. Don't stay with only collagen. Put something more in it. I've been using a lot of different substrates, and different substrates, different results. Put fibroblasts, different results. In terms of of response to drugs, things which are clinically very important. So your model has to be relevant. So try to see what are the questions which are crucial to your project, and from there you go see what are the methods, the tools which are available around. And good luck.